Thank you for the association for having me present the work that I've been doing with my team over the past almost 5 years now. This is an exciting time to think about nutrition because we are almost there to start answering all the questions that you may have. Tonight, I will give you an outline of what you are going to learn. It may not be the thing that you thought you would learn, but I will say that you will learn the significance, challenges, and the value of mechanism-based nutrition research. And I hope to conclude this talk with a sentiment that's very important to me, and it was highlighted multiple times today, is that as a community, how we can support and advocate science. And, the science that I do focuses on, how nutrients in our body influences health and disease states. What you are not going to get from this seminar, sorry for disappointing, is a magic recipe that will cure all of your remedies. Not tonight, but if you come one day and find me on campus, I may give you that recipe. The biggest challenge of our life is really to figure out what should we eat. And, I grew up in Turkey, in the southeastern part of Mediterranean, which is, really obsessed about food. I remember during the day, the only thing that my parents and our neighbors will talk about what to do at night for dinner. It's a very important concept. And so I grew up always thinking and talking about food, and hence I like also cooking. And but the evolution of human nutrition is actually interesting. Because we adapted different phases in our evolutionary history in the ways that we incorporate nutrients. Nutrients, at the end of the day, have chemical information. We eat them, we convert them into biological information. And so here you're seeing basically throughout the course of evolution, started with basically the carnivore phase where we started cooking things on the fire, where we started eating meat. And then when we settled during the civilization, somewhere around 10,000 years ago, we started incorporating grains and agriculture. And then we started seeing explosion in the nutrients and the diets of sorts that you may have heard nowadays, caveman diet, ketogenic diet, high carb diet, high fat diet. It all started somewhere around that time. And now it's, 2023, and we are here. We are either, the world is divided between overnutrition state and undernutrition state, which we call it as malnutrition. And this is really concerning because nutrients have significant correlation and also direct influence on these diseases that I basically summarized here. So every single disease state that you can think of that influences majority of the human population, chronic diseases, non-communicable diseases, infections, cancer, heart disease, neurological disorders, depression, they have some link to the ways that we consume food. And so there are some concerning facts because right now, almost 2 billion adults and almost 2 billion adults are overweight or obese, and almost 500 million adults have undernutrition. The rates for both are increasing. So we are creating a larger and larger disparity. Some part of our society is becoming more and more obese, and another part is having this problem of malnutrition. And then with children's future, this is also concerning because 40 million children are overweight and obese. And it's actually a difficult task to make a children is overweight and obese because of the metabolic demands that they have. But this is happening. There is an obesity epidemic and pandemic both in the US and in the world. And there are almost 200 million children who have undernutrition, and both rates for obesity and undernutrition are increasing. And as I mentioned, the alarm is here. These are not just, individual diseases that you can, stop eating too much or start eating a lot. They bring other diseases with them, and they contribute to increasing rates and worse outcomes in those disease states, including cancer, cardiovascular disease, infectious disease, diabetes, neurological disorders, everything in the spectrum, you name it. So you are what you eat, yes. your grandmother probably told you that as well. But the problem is we don't know how. So when I came to Cold Spring Harbor, in my first presentation, I said, I want to answer that question. It's very challenging, and I will unpack what are the challenges are for you. And thankfully, it fit perfectly. And, it's been a great journey since then. So my lab tries to answer that question. Are you really what you eat? So the nutrients that we consume, they can do many things. They can provide energy for your cells. They can provide building block for your cells, but also they can influence your cell's identity. Like we all have different identities. Our cells also have different identities. And this is by and large encoded in the DNA. But what we are now learning is environmental exposures, including nutrients, can shape the identity of our cells. So that's the complication. Nutrients can influence health and also contribute disease states, but this is not a singular problem. This is a whole body of problems. And so here we try to unpack that problem. So we know that nutrients will influence these cells and how these DNAs in those cells will be utilized to make up cells with different identities that will carry out different functions. And then the complication starts when you start looking at a tissue where it's composed of multiple cells. So in a typical science journey, you do a PhD in a specific field. You are specialized. You get a PhD in immunology. You get a PhD in cancer biology. You get a PhD in neuroscience. And when you look at a tissue, you look at it from that point of view. But to solve that puzzle, you need to understand the whole. And it's not easy. I may say that I want to understand the whole, but do I have the competence and ability to do that? And most importantly, can I do an experiment about it? And will somebody actually fund that experiment? And so the more and more complication starts when you are trying to interrogate organs and systems. There's a big interest in brain-body interaction and how it can influence physiology and disease states. And so in my lab, we study gut and how gut communicates with other tissues and organs, including the brain. But also, this is where things get really complicated. So you may ask me, what should I eat tomorrow? It depends. What's your initial BMI? How old are you? Where are you coming from? What's your genetic ancestry? Do you have it? Are you already sick? Are you super healthy, or are you recovering from an illness? This creates the biggest complication. We exist in a very dynamic organismal state. It's not static. So what can we do about it? So we know something about metabolism and nutrition for a very, very long time. Since those cavemen started inventing different diets, they also started experimenting how different diets may influence disease outcomes. Food was the first medicine. If you read about ancient literature and cultures and mythology, food was the first medicine. And through, generations and generations, these observations led to some associations. And now with empirical evidence, we can establish causality. So we know some correlations that may have some causality. I will give you examples. Ketogenic diets may have some influence on reducing epilepsy. And in some occasions, it may also reduce cancer risk, but we don't know exactly how, and we don't know exactly if it happens in every single occasion. Actually, we know it doesn't. So take it with a grain of salt if you are following a ketogenic diet recipe. Pro-obesity diets is our big concern. We are in Western society, we are industrialized, we have very easy access to food and highly processed food. So that has now my group and others have demonstrated causal, precise mechanisms, how it can increase cancer and contribute to mortality. And then for thousands of years, humans experimented with fasting and dietary restriction, which also has some hints that it may contribute to longevity and health span. And we know little about those mechanistic insights, but not much. And then more on the molecular and cellular side, the stuff that I mostly did in the lab, and I, very excited to basically bridge those two. We know stem cells, immune cells, and cancer cells, they exist in specific states that chooses certain metabolites to consume. Just like you have some choice when you are, in the mood of eating something, when you're listening to music or when you're celebrating something, cells also have those moods that they love eating and consuming certain things. when they are either, at stem cell stage, giving rise to other cells, or when they are differentiated and carrying out specific functions. Same thing with immune cells. If there's an immune cell just resting, it has a specific metabolic demand. If an immune cell is actively fighting infection and proliferating, it has a different metabolic program. And in cancer, that is a even bigger puzzle, because both cancer and the cancer immune fight has some metabolic determinants. So there are some fight over cancer cells and immune cells that makes cancer to thrive in our body and reprogram our entire body and take over, that is partially determined by the metabolic needs of cancer. And so, as I mentioned to you, these two now are close to talk to each other, these two paradigms. And as faster we merge these two paradigms, faster we can come up with good opportunities that can use nutrients as medicine with empirical evidence, like we would know how they would work and we would understand, but also defining the mechanistic targets and use for therapeutics. So the challenges, as I mentioned, human diets have enormous complexity with dynamic variables such as different nutrient types, and the abundance of those nutrients in our diet and pattern. My lab studies fat. There are different types of fat that has different chain length, saturation, complexity, and abundance in the food that you consume. Humans are different. The genetic diversity adds an additional layer of complexity. And here, those two are really where we are limited. We don't know how exactly different diets affect our cells, tissues, organs, and their interactions, and the whole organism. And the reverse is true. When the whole organism adapted to a diet and has an organismal metabolism set, we don't know how it affects our organs, tissues, cells, and molecules inside the cells. And there is a lot of buzz, but we don't even know what we don't know. And that's, like the realization that I want to highlight for a few seconds. As a scientist and as a human being, this is how we, learn things or find truth. So you have an axis that highlights the confidence level about some concept and the competence about that. So I did my PhD in immunology. During that time, I got interested also about stem cell biology. And so I can tell you from firsthand experience, when you are ignorant, you have high confidence about something. But as you start doing it and start gaining experience with failed experiments, wrong hypothesis, then your confidence drops, and then you are like at the brink of giving up. But then something happens as you keep going and keep going and keep going. You reach the bottom of your confidence level, and then you start utilizing the competence and now growing an additional degree of confidence. And so this cycle repeats as you learn new things and you develop new concepts and you try to challenge dogma or you open up new fields. It's very critical for us as scientists to, find new things, but we are stuck sometimes on our path to wisdom in a comfort zone, right? What is that comfort zone? For the things that you are already comfortable with, the knowledge that you accumulated. And so the first principle to me as a scientist, that I can learn anything that's in a textbook. I can learn anything that's published somewhere. That's not difficult anymore because we are living in the information era. What's more difficult is not to sit in a comfort zone and not to take a risk to advance the field. Sit on the horizon of the cumulative human knowledge and ask a question that would advance that cumulative human knowledge. So that's difficult. And for nutrition research, that's very tricky because we have all these information out there. Some of them are not properly designed. Some of them are, probably testing a different thing. It's very challenging for us to get out of that comfort zone and then start a new field. So the wisdom that I hope to show you today is the story of how I was able to, finish a PhD in immunology and then start becoming competent in stem cell biology, and now trying to build different cells and the way that they interact in our body in response to nutritional perturbation and how these things influence health that defines us, and contribute to disease risk. So my solution to that is basically to choose different models that can talk to each other. So we study epithelial stem cells, immune cells, and microbes and how they talk to one another. And we use diet and diet-induced perturbations such as obesity to probe these individual networks. And then we study them in the context of a disease, which helps us to define mechanisms. And then with what we learn, we can apply them to physiology. And the physiology that we study in my lab is tissue regeneration. So we study these things in cells, in tissues, in organs and systems, and in organismal states, as I mentioned. And so we take these cells individually, we grow them on a dish, we can study how nutrients affect them. We can add different cellular structures to one another and monitor how they are talking to one another. We can make genetic models, genetically engineer the mice to turn on and off certain cells and genes and then monitor them. Then something very exciting that I was able to do at Cold Spring Harbor is design new diet types smartly to study how different nutrient types influence health and disease outcomes. And most importantly, what's the human relevance? And this is the partnership that we have with Northwell Health, where we were able to develop comprehensive human biobanks for us to able to study nutritional states and also pathologies such as obesity and also in the context of weight loss, the contribution of metabolism to cancer states. So this is one of the most comprehensive biobanks that I know to study obesity and obesity-dependent alterations. And we were able to so far collect more than 500 patients in that biobank to study the functional consequences of some of these mechanistic discoveries in a human setting. This is very, very valuable. So the simple journey of food in your daily life, it starts here, you chew, then it goes down to stomach, and then the absorption takes place in small intestine, and then ends in large intestine, and then you get rid of it as feces. And so when you look closely, this tube of intestine is composed of these structures. So in the small intestine, there are those villous finger-like structures that allow the absorption. And at the bottom of that structure is the crypt, which harbors the stem cells that regenerates the tissue. So we study these stem cells at the bottom of that crypt to understand how regeneration occurs and how these specialized cell types carry out gut functions. And we know which molecules they need to do that. So we can grow them on a dish and study basically what's happening in an organism right on a dish by looking at those cells under a microscope. So this green is a marker that we use to trace these stem cells. And so these stem cells or epithelial cells are not in isolation. So we need to understand how different perturbations can affect neurons, fibroblasts, immune cells, and microbes, and how they communicate in that little crypt area. So I will give you one example how we can do this in a modular manner that will make an impact on human. So the first example builds upon what I did a little bit during my PhD and continued to do here. So we took the challenge on understanding why obesity is a risk factor for cancer. One of the cancer types that shows high association with obesity is colon cancer. And so we developed these mouse models where we used the pro-obesity Western diet, gave it to a mouse, and then found out that after 9 months of feeding, chronic feeding of this high-fat diet, these mouse developed spontaneous tumors. And we found the reason that these mouse were developing tumors is because their stem cells became hyperactive stem cells because the fatty acids in the diet was activating something on the nucleus of these cells called a lipid sensing factor that's reprogramming those cells, and they are becoming now hyperactive. So this hyperactivity we found contributes to the increased risk of cancer. Now I can tell you in this defined concept why if someone consumes a lard-based high-fat diet and becomes obese, through which pathways the cancer occurs. So then we use that new knowledge we learned that fatty acids is doing something to stem cells, and they are becoming hyperactive, and when they get a mutation, they become cancer. But there is a value here to apply for a regenerative application. And this is what we did. We developed regenerative diet regimens by looking at different fatty acid compositions. In this context, not in a pro-obesity Western diet, but a regular diet. We started engineering diets by learning from the mechanism. And what this allowed us now to figure out that, yes, the regenerative fitness in the tissue can either lead to cancer if it's hyper-regenerative, or if it's not regenerative at all, it can lead to diseases like mucositis, inflammation, wounds, and aging. And so, but there is this sweet spot here where we can use a regenerative diet regimen and make resilient stem cells. So in our work, these regenerative diet regimens protected intestinal epithelium from radiation damage, from chemotherapy-induced damage. And this is remarkable because if you talk to any cancer patient who's receiving chemotherapy or radiotherapy, one of the most debilitating side effects is GI, gastrointestinal side effects. And we believe that this, coming from this discovery, has a potential to help patients. And we started our path towards a clinical trial. We are working on figuring out how these dietary regimens will work in human. But this is very remarkable. This happened in the past 4 years. So I will conclude this section by saying that your stem cells are what you eat. So the second part is very exciting and more complicated, but because we are dealing with now this time cancer and immune system. So as I mentioned, tumor cells and immune cells are in constant fight between each other. Immune cells are trying to eradicate the tumor. Tumor cells are trying to suppress immune system. What we realized, and many people actually in the field now are realizing, that there is similar metabolic demands and programs in cancer cells and immune cells. And this metabolic tug of war between cancer and immune cells is very important for us to understand so that we can develop therapies for that. And then getting more complicated, now let's look at 3 different components. This is something that I wanted to take on the challenge and show myself and the field that we can do it. Look at different modules. So microbes, stem cells, and immune cells. We used the model of pro-obesity Western diet because it's such a harsh perturbation to measure what's going on in the microbes and immune cells. What we found was the fatty acid in the diet is perturbing the microbes. Certain microbes are getting lost. And also immune cells. Certain immune cells are losing their fitness. And because of that, there is a miscommunication between immune cells, microbes, and the ability of the immune cells to survey those tumor-initiating stem cells. Every day, one of your cells will have a mutation because of problems in DNA replication and repair. But your immune system is actually very good at seeing those cells and destroying them. However, in this context, what happened is because of this pro-obesity diet, and we believe it's driven by these fatty acids, this communication is perturbed and these cells are escaping and forming the tumor. So now we want to ask a very difficult question. Does this occur in other diet modalities? So we wanted to deconstruct and reconstruct that complexity. To do that, we opened a Michelin 3-star mouse restaurant. If you have rodents in your household, send them. We can, don't send them actually, we cannot do that. But we developed basically different diet regimens for mice, highlighted here in different colors. So we made the different fat-containing diets ranging from lard, palm, olive, milk, coconut, fish, and a ketogenic lard. And then this is a longitudinal study that's been going on for 3 years now, and we finally have all the data and analyzing it. And so you can see that we did a longitudinal monitoring of these mice and assessed how they respond to these diet perturbations. And we also did reversal of the diet because humans, when they realize that they're doing unhealthy, they immediately go and change their behavior, change their diet, thinking that that is going to save them. So we reverted at 4 months and also at 9 months. And so I want to just highlight one discovery here is that we found that while the weight loss is successful in these mice after reversal to control diet, the microbiome perturbations persist after reversal to control diet. So even if mouse goes back to the baseline in terms of its weight and some of the metabolic measures that we did, there were some persistent changes in microbiome, in immune cells, and in stem cells. I want to highlight the microbiome because this is very interesting, and I'm sure most of you will have some questions about this probiotic and that probiotic. So these are like commensals, lactobacillus johnsoni, and B. faecalis. So these are probiotics that help our immune system to get educated, especially the lactobacillus johnsoni. But what happens with this high-fat diet perturbation, and especially I highlighted here with coconut, ketogenic, and milk fat, you see this dampening that doesn't come back after the reversal. So the conclusion here is your microbes are what you eat, but even after you change your diet, the effect can persist. So finally, this is outside of my comfort zone, but because we built those models, I started talking to neuroscientists. So at Cold Spring Harbor, we work with some neuroscientists to interrogate these different diets in neurological disorders. But in this study, which is really remarkable, I collaborated with a friend of mine at South Carolina who develops this repetitive mild traumatic brain injury models. So this model is similar to the NFL players. So they get brain injury, but it's not very harsh so that they don't get hospitalized, but it's mild, but it accumulates. So early on in the life of these mice, we gave them these mild injuries, and then we let them either go on a control diet, or we alternated the fish oil diet, thinking that it is gonna make it better, because why not? Fish oil is great for you, right? So we gave these mice 2 days control diet, 1 day fish oil diet, no major change in metabolic alterations systemically. But when you look at the brain, and this is an electron micrograph picture, I don't expect you to read it, but just watch it. So this is a healthy brain. And this is what happens to a brain 6 months later. There are some mild degenerative features you can see, but because this is a repetitive mild injury model, you don't see the ramification that much 6 months after that injury. And in the fish oil, without the injury, this is a healthy brain, it's electron microscopy image. And this is the stark contrast that I want to show you. There is no structure here. It's all mushy. And the reason that we believe is that because this fish oil contains these reactive fatty acids, and after an injury, for some reasons that we are still trying to figure out, it messes up with the ultrastructure of mitochondria and the cells and their extracellular matrix. So this is, I wanted to show you as an example, because context also matters. You are what you eat. Your brain is what you eat. But contrary to what you think you may know. So, I always study the gut. I know fish oil and milk fat can have some beneficial effect, but we need to really understand the specific context in which we are trying to interrogate nutrition as a remedy. So I will just highlight this, that we have this course throughout the evolution. We are here in malnutrition. We are at risk of many diseases, but with mechanism-based nutrition and this type of research, I hope that we will make a difference. And in my lab, the studies are aiming to define precise nutrient regimens that modulate human health and mitigate disease. We really want to develop these data-driven models that can predict potential causal mediators, and then we can interrogate them in considering human genetic and exposure complexity. And then these mechanistic targets, in the work that I didn't describe today, we are actively working on them to develop therapeutics. So you can define a gene, you can define a protein, and go after it and make biologics independent of the nutritional intervention. So your future is what you eat, and hopefully we will make your future healthy. But I want to end with this. And this is very important to me because before coming to Cold Spring Harbor, I did my PhD at Harvard, and it's an amazing place to do your PhD. But when I came to Cold Spring Harbor, I was amazed. And I called my wife and I was like, I found my place, because it really values science. It's a science-first mentality place. And so this Foundations for Future campaign is very important for young scientists like I am, because I can't sleep at night because I'm so excited about these discoveries that we can make. And these discoveries are based on these three pillars. You need to always accumulate knowledge. That's intellectual aptitude. Cold Spring Harbor is an amazing place because we have meetings and courses. I can attend any meeting at any subject that I want to know to increase my intellectual aptitude. Critical thinking. You need to discuss. You need to critically evaluate your own beliefs. We don't have any departments. If I talk to a person outside of my field, will give me the most pure and honest opinion, devoid of any jargon and comfort zone that the field is in. And then the effort. We can do experiments that are risky. We can take a leap of faith and go after something to slay the dragon. So that happened in human history a few centuries ago. And, Enlightenment era generated basically all this scientific foundation that we are currently standing on the shoulders of. However, as I mentioned to you, our society is also divided, right? So science and society has some issues, and we need to make sure that we bring back science and society for a new renaissance. And for Da Vinci, there was the Medici family, and you are the association and the board and the trustees. You are our family. And I would like to, on behalf of everyone at the lab, my team, my colleagues, thank you for your continued support. And I hope we will be able to make impactful discoveries in the years to come. And with that, I would like to thank the team and the colleagues that I have that, made this work possible.